All right. Buongiorno a tutti. Good afternoon, everybody. What can I say after Steve? He's a rock star. So I'm going to talk about a different angle, slightly different angle, which is inflammaging. And I'm also going to allude to the work we're doing in space biology to accelerate aging of different blood samples and specimens and organoids, and have a window for that accelerated aging and see if we can identify interventions to reverse that. I'm a director of the 1000 Immunomes Project. My background is in human immunology and data sciences. And I'm sure you know that the immune system changes with age. There's drastic changes in cell proportions, in inflammation, and that's part of my research over the past 18 years. So I'm just showing you here one subset that changes a lot in blood. That's the CD8 T cell memory cells. When you use more cells, looking at different proportions, you can build clocks. And this is one of the immune clocks that we have built using different immune cell subsets. And these cells can actually infiltrate in different tissues and organs and cause pretty bad consequences. This is work from Patricia Noon at Stanford, who took cardiac tissue from postmortem individuals, and the arteries in plaques were full of T cells that were actually activated, clonally expanded, and recognizing some things there. And so not a really good endpoint here in terms of the pathological consequences of the immune system in heart. A more general and broader effect, as you know, is inflammation. I think we all understand now that inflammaging is a big deal for longevity. It's been all over the news, and we published this paper demonstrating that really this comes from the exposome. So what we eat, how we treat our bodies, the level of chronic stress, what we breathe, the quality of the water. A couple of days ago, I posted on LinkedIn something related with plastics, nanoplastics that are infiltrating and accumulating in tissues and raising inflammation and causing very, very bad effects in every one of us, really. But so there's a lot of prevention that we could do. And this paper was published by one of my team members that got the cover of Molecular Metabolism, really pushing the idea that chronic inflammation accelerates, and it's at the center of each one of the hallmarks of aging. Probably till 4 or 5 years ago, there were no accurate biomarkers of inflammation. If you look at you cut yourself and you develop acute inflammation. That's the cardinal signs. It's red, swollen, and there are canonical markers, interleukin-6. These are different cytokines. For systemic chronic inflammation, there's really no good markers. So we set out to establish the 1000 Immunomes cohort almost 17 years ago at Stanford. And this is a large study in 1,000 people that were followed over the years. We recently secured another $18 million to extend this project for the next 5 years. And we've measured a lot of different things, sequencing, proteomics, metabolomics, and from all the data, it became pretty clear that proteomics was very informative for different diseases, for different conditions, and so we decided to use a nonlinear approach. This is the deep learning approach, so AI type of nonlinear approximation to this to create an inflammatory clock of aging. And this predicted multimorbidity, as shown in the top left, frailty in older individuals, immune function, ventricular remodeling, exceptional longevity. So in individuals 100 years and older, this was very low. And when we build a gene expression surrogate of inflammatory age, we can also predict mortality in the Framingham Heart Study. So one of the theses that we had in my company, so we brought this to the marketplace, is that lowering inflammatory age may have positive health effects. And so we came up with a stack of different computational methods to identify interventions that may work at lowering inflammatory age for those that have elevated I age. So how do we do this? We take different compounds from the public domain. These are drugs, supplements, natural compounds, around 120,000 compounds, and we build gene-chemical interactomes. And these are 1.6 billion interactions. And then we ask the algorithm to pull compounds that will work for that particular I age. Okay, so we found drugs and different compounds that can do the job at lowering inflammatory age in silico. Then we go ahead and can map those to foods and food servings. And we identify a particular food that was rich in beta-glucans and rich in phenols, and that one actually decreased inflammatory age in people with high inflammatory age at baseline. So this is a study we did with a corporate partner, and giving them in 2 arms. One is placebo, one is the experimental group, and as you can see here in this plot, inflammatory age was down as soon as 2 weeks after consumption of this rich phenol and beta-glucan compound, and also the protein eotaxin, which has been associated with neurodegeneration, went down significantly in this group, and that was not really true for the placebo group. So we decided to go one step forward and create personalized interventions. So the one-size-fits-all was what I just showed you before. I could have an inflammatory age of 40, and my neighbor could have the same inflammatory age, but the way we get there is different. I could have elevated protein A, and my neighbor could have elevated protein B. So there is a world where we could develop personalized interventions. And that's what we did in my company. 750 participants, half of them treatments, half of them placebo, and we measured in 4 different time points, different omics, multi-omics, inflammatory age, all the proteins. We took photos of the face and the scalp. A recommendation for those doing studies of any sort in humans, take a photo of the face, and I can tell you why in just a moment. We also collected clinical endpoints and a large survey encompassing different things. It's a long 4.5-hour survey that contains information about clinical parameters and also exposome parameters. So the study is still under analysis, but one thing that I want to allude to is that we developed this photo age. And now what we're trying to do, this is pretty easy. Okay, there's a lot of different algorithms that can predict the age of someone using a photo. That's a pretty straightforward computational tool, mostly based on wrinkles and stuff like that. What we want to do is to take this photo, pair that with sequencing data, and be able to predict, based on the photo, your organ aging. That's what we call the healthy selfie. And it's in collaboration with the Buck Institute. To complete this, we need a couple $100,000. So if there's philanthropic donations, are very welcome, and we can talk about this further. I was predicted to be 37, so I gave a bonus to my postdoc. I'm 48, so that was really good news. I was asked to be controversial here and try to ignite discussion. So I would like to ask everybody in the room, think about it for a moment. Do you think, and just raise your hand if you say yes, that old age should be considered as a disease? Think about it for a few seconds. Oh, wow. Nobody out there. There's one. Yes. Thank you, Steve. Okay, so this has caused several, it's probably 5% of the room. This has generated a lot of debate in the aging space. Many folks think yes, others think not at all. But the resolution to this, I think, came recently because the WHO, after several rounds on trying to recognize old age as a disease, they said healthy longevity is a process of developing and maintaining the functional, that's important, functional ability that enables well-being. We've talked about well-being, I think, over the past week in this conference. What are we referring to here? If I like, as an individual, I really enjoy reading books, I like to be able to read books until I'm 100 years old. If I like go surfing or hiking, I like to do that until I'm 90 and until the last day of my life. That's the idea here. And so they recognize for the first time the loss in intrinsic capacity, which is another version of function, in the International Classification of Diseases, ICD-11, under the code MG2A. And so functional aging really declines at the very last stage of probably last 20 years of your life. The first thing that starts to drop is biological aging at the age of 30 or 40. That follows phenotypic aging. And then last thing that changes is functional aging. And I'm going to give you a few examples. I'm kind of running out of time. No, I think it's good. Something's going on here. Okay, good. So just to explain what I mean by functional aging, here we have biological aging, all the biomarkers of aging, all the hallmarks of aging that we know, protostasis, oxidative stress, inflammation, so on. Phenotypic aging will be loss of muscle mass. I do a brain scan and I see that the gray matter volume shrinked. But you still are healthy, you still look good in terms of your cognitive capacity. And then functional aging is when you start to lose capacities, functional abilities, intrinsic capacity. So frailty, geriatric syndrome, loss of cognitive capacity, and so on and so forth. And there are well-established metrics to measure those things. The psychological, cognitive, vitality, sensory, and locomotion systems are captured now by an app that's called iCope that was developed in collaboration between the Toulouse group, Bruno Vella there, and the WHO, where you're using this in 45,000 people with Toulouse to understand biomarkers of functional decline and intrinsic capacity. So I'm showing you here an example of what? Phenotypic aging. This is gray matter volume in the brain. This is a collaboration with Joel Kramer at UCSF. Every dot here represents an individual scan, and things change with age. There's a huge amount of variability, right? And there's also here some functional aging. So this is mild cognitive impairment in red and pre-mild cognitive impairment in yellow. All cross-sectional data. It looks great. Now, it may be predictive of bad things that happen, dementia and stuff like that. But there's a huge amount of variability. And I want to stop there for a moment and show you that trajectories are more important than single time points. So here we built yet another clock. There's a lot of clocks out there, and we build clocks for a living, really. And I'm showing you the first visit of the Stanford 1000 Immunomes cohort. And what we did here is to build the immunoproteomic age of cardiovascular etiology using blood samples. And you can see this person here is, one would say, is accelerated, is at risk. And this individual may be protected. Now when you look at changes over time, trajectories, that person that was up in the curve actually maintained his levels and didn't develop cardiovascular disease, whereas this one here had this accelerated aging. So there are many, many cases in which single time points are not informative, and we need to follow people longitudinally to have a better idea of what's going on there. That's what we're doing in my second company. I want to show you some of that. This goes back to, I'm going to switch gears a little bit here. So this goes back to a relationship I started with Susie Sanello at the Human Research Program in Houston. She reached out to me in the first place because she said, David, the research you're doing in the immunome and aging is very well aligned with what we do to protect our astronauts for the risks associated with spaceflight. That blew my mind. I didn't know anything about space biology at that point in time. I was excited because I always wanted to be an astronaut, but that's a separate story. I didn't know that spaceflight accelerates aging. Astronauts have 5-fold increase in developing cardiovascular disease over the life course. They have cognitive deterioration. They have reactivation of viruses, immunosenescence, immune decline, inflammation. Very similar to what you see in aging, but at an accelerated fashion. For each year these people spend in the International Space Station, that corresponds to 30 years aging on the ground. That doesn't mean they go back and they'll recover. For the most part, for soft endpoints, they do recover. But for harder endpoints like ventricular remodeling, stuff like that, it may take 5 to 10 years to recover, okay? So Susie passed away in December of 2023 of a terminal pancreatic cancer. So really, I'm taking this as a mission in my life. Many of the things that we discussed with Susie were around how space research has contributed to our daily lives here on the ground. Things from information technology, satellites, your cell phones, consumer goods, all the way to health and medicine, and that's our focus today. So solar panels, Velcro, baby food, formula, et cetera, et cetera. So I mentioned that space causes accelerated aging in different organs. Well, it's mostly because these people suffer from low gravitational forces. Not so much the radiation issue. They're protected from radiation in the low Earth orbit. If you go above and beyond LEO, then you get galactic radiation, and those really, really bad, a lot of DNA damage there. So we're using and pivoting on that reality and envision a way to simulate microgravity on Earth in the lab, okay? So we're using 2 instruments, one that was developed by the European Space Agency, is shown in the left, one that was developed by NASA, and by doing so, we take cells from individuals, we also take organoids from people, and we can develop those in the lab, expose them to microgravity, see how they age at an accelerated pace, and then test a number of compounds to be able to prevent or reverse that aging process from happening. So this is personalized precision medicine, or more so longevity, precision longevity. So we just got our paper accepted in Nature Communications, and the workflow works like this. We take a blood sample. In 25 hours of simulated microgravity, we can age the samples by about 10 years, 8 years, 5 years, depends on the individual, it depends on the tissue. There's tissues that are more sensible, more sensitive, like brain organoids, and there are tissues that are less sensitive. So here I'm showing you one of the methylation clocks that we measured. That's called the Dundin pace of aging. Unfortunately, there's a lot of brightness here, so you can't see the connecting dots, but some of the people, most of them, increase their methylation age. Some of them stay the same, so there's that room for personalized understanding of what's going on. Inflammation goes up, cellular senescence, signs of cellular senescence goes up as well. And then we use a very similar approach than those that I showed you before for edifice. We pull a number of different compounds from this in silico screening. And what I'm showing you here is the potential therapeutic compounds from Drug Bank, Links L1000, a large repository from NIH, and FoodDB that has all the food ingredients and natural compounds there. And then the chances or certainty that these different compounds will reverse the microgravity-induced aging genes there. We test these compounds in the aging cells in the lab. And what you can see here is the efficacy of these predictions, 68 to 70% of the reversal of the differential expressed genes induced in microgravity. Just one example that a IH index goes down significantly in every one of the individuals tested here. So as a summary of that workflow, we can analyze the next 5 years of biological aging using our technology. We do compound screening in silico. We select the top compounds, and then we test those in your aged cells. So we have a window. We age you to see what will work, in other words. So it looks great in blood. What happens in more complex tissues? I mentioned cardiac organoids, brain organoids are more sensitive to cardiac ones. We bought these from vendors. We can also develop those in the lab. They come from, these in particular come from iPSCs, induced pluripotent stem cells, but we can also do direct differentiation, right? You can take blood and direct differentiate your blood into a muscle, into a neuron cell or a cardiomyocyte or an adipocyte. And we can do this and test this in our platform. And as you can see here, in all cases, or mostly all the cases, this is cardiomyocytes alone, cardiomyocytes with endothelial cells, and brain organoids. This is really an impressive technology. Those hearts, mini hearts, are beating, are completely functional. The brains can actually learn, believe it or not. These are mini brains, 3 to 4 millimeters in diameter, and they age by about 10 years when we expose them to microgravity conditions. So just to reiterate, we can predict how that person will age and prevent those mechanisms of aging. This is part of my company. As I said, it's called Cosmica Biosciences. We are in South San Francisco. And this is how our report will look like for those interested in concierge medicine, or they have your own longevity practice. This is how the report will look like. On the left, you'll see how fast you'll age in the next 5 years. Right in the middle, you see which hallmarks of aging will affect you, which may be very different of those that will affect my aging. We have different disease risks. The top compounds that came from our silico testing, we can test those in vitro. And last, we have something really interesting that came from a meta-analysis we did, in which we can take all the lifestyle interventions that have shown effects in different hallmarks of aging from the literature. And then we can say, okay, if this person will have senescence, what can we recommend in terms of longevity interventions or lifestyle changes? So with that, I'd like to thank everybody for your attention, and those are my contact information. Thank you so much.